everybody, coming at you from my office. Now we're going to talk about the urinary system, which is also known as the excretory system or the renal system. This is basically how we excrete waste products from our body, except for CO2. We use the respiratory system to do that. But you'll see that the kidneys actually do a lot of different things. So it does maintain the water balance in our body. So with that, remember that was homeostatically controlled. So if we do have too much water in our body or not enough, the kidney can help modify that by either excreting the extra water or reabsorbing precious water so we don't lose it from our body, right? It maintains the extracellular volume and also the osmolarity of the body fluids. So it just balances out the salts and glucose and everything else, which will then also affect our water balance. Some of these seem to be a little bit redundant. Again, the concentration of ions, the electrolytes, we can balance that with the kidneys. Help to balance the acid base in our body as well. And the next lecture will actually go a little bit more detail with how several body functions do that. And of course, we need to excrete several compounds from the body. And since we do this through filtering the blood, there's a lot amount of blood that passes through the kidney. And so when it does that, we'll see that we actually have structures within the kidney that can determine the number of red blood cells. And therefore, if our red blood cells, if we're low, then it can stimulate the excretion of, or excuse me, the secretion of a hormone called erythropoietin. Right, and so erythropoietin is a hormone that can then signal the bone marrow to produce more red blood cells. Okay, we also produce renin, which is another type of hormone that's going to affect sodium levels, and then it helps to convert vitamin D into its active form. So we're going to go through many of these. But not all of it. But it's the main route for eliminating toxic metabolic wastes and other foreign compounds from the body. That's one of the main things that it does, and maintains that proper water balance. So our kidneys are, we have a pair, and they're located on the back wall of the body. And you can see that we have an artery that branches straight off of the aorta into each kidney. That's the renal artery. And then as the blood flows around in the kidney, comes back through the vein, then that's the renal vein that connects right to our inferior vena cava. So we quickly cleanse the blood quickly and put it back into into circulation. And so from there, within the kidney, once it makes urine, it flows down the ureters, and each ureter connects to the urinary bladder. Okay, and then we can store the urine there until we're ready to excrete it from the body. And then it exits through the urethra. Alrighty, so that's just kind of the general overview of the whole organ system. Remember that the adrenal glands sit on top of each kidney, but they don't really function in this excretion. But we will see that some hormones that come from the adrenal gland do affect the functioning of the kidney. So that'll come later, right? And all of this is right below your diaphragm. Now we have a pair, but you can live with one kidney. So one kidney seems to function just fine with cleansing the blood, but you do need at least one kidney. So here's the opening of the kidney. So some of this, if you've had anatomy, will be a review. This is basically when we open up the kidney, we can see that there are two main areas associated with the kidney, and that's the outer cortex and then the inner medulla, right? And the medulla is mostly separated into these renal pyramids. And so we're going to see that the functional unit of the kidney, the nephron, actually spans through both the renal cortex and the medulla. At the end of each renal pyramid, though, the collecting ducts, where the final passage of urine is from the nephron, that actually then gets collected into these funnel-like structure is called calyces. So one minor calyx will merge with other minor calyces and form a major calyx. From there, these major calyces merge into the renal pelvis, and then that's like a giant funnel collecting all the urine down to the ureter. So here's mainly what I just said, and you can see in a little more detail the cortex and the medulla. And then here, though, we kind of blow up a little section between the cortex and the medulla. And now you can see the functional unit of the kidney. And these are these nephrons. And so the nephrons are the tubules where the fluid from the blood is received, the filtrate, and then as the filtrate passes through these tubules, it gets modified. So again, we conserve water or we release water, we conserve salts, release salts, whatever it is that we need to do passes through. And then this is going to be associated with that blood supply. So remember that the blood supply comes in not only to filter the blood, but then many of the products we actually reabsorb back into the blood. And so capillary beds will be surrounding all of this as well. Okay, so because the nephron is the site of blood filtration and then modification, this is what's known as the functional unit of the kidney. This is the structure where these function of the kidney is coming into play. So there's approximately 1 million, it's like 1.25 million nephrons in each kidney, right? And so it really has two components. The nephron itself is that tubular component, which is what you can see here. So think of it as a big straw where fluid enters, and then again, as it moves through the straw, the walls of the straw are actually made of cells. So these cells go to work to either reabsorb water, salts, secretion, all of that kind of stuff. And we'll get into all of that in a minute. And so the tube is one continuous tube, but it has these functional regions. So again, the cap that kind of surrounds the capillary where the filtration takes place is called the Bowman's capsule. And then the first portion here of the tube is called the proximal tubule. And then we move from the proximal tubule, you can see it twists and turns, and then it forms this loop that goes down. So all of this up here was in the cortex. The loop goes down into the medulla and then comes back up. So the whole thing is called the loop of Henle, but we're going to see that each side of the loop has a different function. And one side then will be called the descending loop because it goes downward. And then as it turns upward, it's called the ascending loop. And then as we get back into the cortex, the tube kind of twists around again. Now this is called the distal tubule. And then that's really the end of the nephron, because then a distal tubule from different nephrons can each merge into a collecting duct. And so this is the urine then that will drain into the renal pelvis and then to the ureter, and we hold it into the bladder. Okay, so that's the tubular portion. We also have the vascular component. So again, remember that we have the artery and the vein that branches throughout the kidney. Again, if you had anatomy, you learned about the names of all those blood vessels, right? Well, we're mostly concerned about the last arteriole that branches into the capillary bed. So that arteriole is called the afferent arteriole. And then it branches into the capillary. It's like a knot of capillaries called the glomerulus. This is where the filtration takes place. There's not a lot of gas exchange that takes place here. So at the other end of the glomerulus, or that capillary bed, is another arteriole. This is called the efferent arteriole. But you'll notice then that the fluid, the blood that doesn't get filtered out, that remains in the capillary, then exits the efferent arteriole and then forms these peritubular capillaries that span all around the nephron, the tubes, right? So when I say that the the tube, the nephron will reabsorb. So something that's in the filtrate will leave the nephron. It'll get into the peritubular capillaries. And so it's back into our blood supply, right? So that's how we reabsorb things. And that's, you can see that that peritubular capillaries merge back into our renal vein eventually, again, into our inferior vena cava, okay? And so we're going to look at the actions of all of these blood vessels and the tubes and how we produce urine and what this nephron is actually doing. Okay, so so the nephron, there's really kind of two types of nephron, and here's a better picture of showing the difference of the cortex region and the medulla. And so some of our loops of Henle are relatively short, and you can see there's a short projection down into the cortex, and most of it's up into the, I'm sorry, projection into the medulla. Most of it is up in the cortex. Whereas down here, these are longer loops of Henle, and this type of nephron is called a juxtamedullary nephron. Okay, and that's because the loop of Henle projects deeper into the medulla. And that's significant because we're going to learn that the loop of Henle is the major site of water reabsorption. And the longer the loop is, the more water that can get reabsorbed. And also the medulla, the interstitial fluid, is very salty. So that sets up a gradient, a hypertonic gradient. It's hypertonic outside, hypotonic inside. So water is going to move out toward the medulla, and of course those capillaries are there, so they'll pick up the water. Whereas you can see here with a much shorter loop of Henle, you're going to get a more limited supply of water being reabsorbed. Okay, we humans have mostly cortical nephrons. We do have some juxtamedullary nephrons, but if you were a desert dwelling animal, like a desert rat or something, you actually would have a lot more of juxtamedullary nephrons because you don't have very quick access to water. So those animals have to conserve as much water as possible and not urinate quite so much. They still have to urinate a little bit because we need water to help move the waste product out of the body, but they don't urinate nearly as much as a human would. Okay, so let's look at the first segment and the first function, the blood coming in and the filtration. Actually, let me go through all the functions. I almost forgot that. This is just a simplified view of a nephron and different parts of the tubule, right? And so we shortened it so that you can see these three basic renal processes. So again, it has to start with filtration at the glomerulus. So we refer to that as glomerular filtration. So again, blood's going to come in the afferent arteriole. It's going to be under pressure, and that's going to cause the fluids from the blood to be pushed out of that capillary into the Bowman's capsule here. Okay, and the rest of the blood that remains in the capillary is going to exit through the efferent arteriole and then become part of the peritubular capillary. Okay, but what we'll learn is that when we filtrate the blood, we do it indiscriminately. We don't seem to have the ability to pick and choose what gets filtered out of the blood. Okay, so there are good things that get filtered out of the blood. So we're going to reabsorb. Okay, so TR stands for tubular reabsorption. Okay, so you have to remember these definitions and which direction fluid is flowing. So in filtration, fluid is flowing from the glomerulus into the Bowman's capsule. And then all that fluid now inside of the nephron is called filtrate. Okay, but now we're going to manipulate the filtrate. And some of the items that are within the filtrate, we're going to put it right back into the capillary, right? And the peritubular capillary portion. So that's called reabsorption. So movement from selective movement from the filtered substances into the capillary is reabsorption. Okay, but remember, we don't filter everything. So there are some things left in the blood that we may want to get rid of, right? And we missed our chance at the filtration. So later in the nephron further away, we'll have the ability to selectively secrete items from the peritubular capillary into the nephron, and then it becomes part of the urine to be eliminated. Okay, so we're going to look at each one of these processes and see what part of the nephron that occurs in. If I can get my mouse to work so I can advance the slide. There we go. All right, filtration. So a couple of things we need to look at with filtration. One are the barriers in filtration. Okay, so remember, fluid has to pass through the capillary, through the cell membranes. Here's a close-up of the, well, here's a capillary here. The inner lining, the red dots, indicate the epithelial lining of the capillary. So this is the lumen of the capillary here. So as filtrate runs, blood runs through here, remember that our capillaries are leaky. Well, they're really leaky in the kidney. They're even more leaky. You have these holes in there called fenestrations. So blood is under pressure, right? So that pressure, hydrostatic pressure, pushes the blood against the walls of the capillary and things squeeze out through these little holes, right? So that's one barrier, okay? The next barrier, you see number one there, the pores between the endothelial cells of the glomerular capillaries. So again, capillary pore is also called fenestration. You can use either term, okay? But remember, one of the properties of epithelial tissue is that it always has a basement membrane. Remember the basement membrane? And that's full of like proteins and glycoproteins, sticky stuff. So the filtrate has to, the plasma has to also pass through there, okay? And so that's a second filtration barrier. So some things can move through the glomerular wall, but things like red blood cells can't get through the pores, right? So those stay behind. But maybe plasma proteins start to exit through the pores, but they get trapped by the basement membrane. So they end up getting swept away back with the plasma in the blood, right? So they can't get through. So that's the second layer, is the basement membrane, okay? The third layer are these cells that lay over the top of the glomerulus. So you can see all these little branching structures here. That's actually a cell, okay? The cell is called a podocyte. Okay, and those podocytes are derived from the cells of the Bowman's capsule. So look at the Bowman's capsule. It's made out of simple squamous epithelial tissue. And as it comes around to where the entrance of the afferent and the efferent arterioles are, it wraps around, and now those epithelial cells wrap over and kind of grab around the capillary, right, of the Bowman's capsule themselves. Okay, and so now we call those cells a podocyte. And those podocytes, they have these cytoplasmic extensions. The cells just extend out so that they can grab and wrap around. But notice on my fingers here, I have space in between my fingers, right? That's the space where then this fluid that gets past the capillary, past the basement membrane, and now it can pass through that space. There are these little slits, okay? And so that's the third barrier. So some things can get past there and other things can't. Okay, so I put a video, a Khan Academy video on Canvas that kind of steps through these three barriers. So it is important to know about the three barriers. Okay, let me show you the next picture because this will show an actual podocyte. There it is. There's an actual podocyte. Okay, again, it's like an inner layer of the Bowman's capsule. So it's like a visceral layer of a tissue. Remember parietal and visceral? So if we go back one, the Bowman's capsule cell itself is like the parietal layer. And then this tissue, or the cells surrounding the capillary, that's like the visceral layer. Okay, so again, those are the podocytes. Here's the cell body where the nucleus is, and then the cytoplasm just kind of pushes out the cell membrane and forms these branches, which form even more branches. So it's in between these slits that are the filtration sites. So that's the third barrier. All righty, so I'm gonna say it again, 3 barriers of a filtration site, right? The pores between the endothelial cells of the glomerulus, the basement membrane and the filtration slits between the podocytes. Okay, what's interesting is in podocytes, they found actin and myosin in there. So there's no muscle, but actin and myosin are there. And we think we can manipulate the size of the slits by that sliding across. We can kind of pull and contract and control the size of the foot processes, which will control the size of the slits. So we don't know how we do it yet or if we do it, but maybe we do actually control what gets filtered. But that's just kind of an extra, don't worry about it. Just interesting. Okay, so now we know about the three barriers. So I mentioned that the blood, of course, is under pressure. So it's really pressures or forces that cause the filtration. Okay, so we have to consider there are three different physical forces. Some of them promote the movement of fluid from the capillary into the Bowman's capsule, but some of them oppose it, right? So just like we saw filtration and reabsorption in other capillaries, we need to look at those filtration pressures and find out which ones overcome the other ones to determine the net filtration rate. Okay, so the pressure inside of the glomerular capillary, right? That's blood pressure. That's the fluid exerted on the blood. That's that hydrostatic pressure. Well, when it gets to the capillary, it's about 55 millimeters of mercury. That's just something to remember. Okay, and so that, again, that pressure is a hydrostatic pressure pushing against the walls of the capillary out. So that influences pushing fluid out. Right? So now, but inside the capillary, right, we have plasma proteins. Remember those influence that's the plasma colloid osmotic pressure, right? So that wants to draw fluid back in. Okay, so there's a plasma colloid osmotic pressure inside the capillary of about 30. So again, that opposes filtration. That wants to pull the fluid back into the capillary, right? Then the Bowman's capsule has fluid in it, so it has a hydrostatic pressure. The hydrostatic pressure, again, is pushing from the Bowman's capsule out toward the capillary, right? So that also opposes filtration. So we have two forces that oppose filtration, okay? And the Bowman's capsule hydrostatic pressure is at 15. Okay, so what we need to do is we need to add up these pressures. Those that favor filtration will be positive. Those that oppose filtration will be negative. And so I did write, again, if I can get my mouse to work so I can advance there, I'll do it this way, All right, let's do the math. So here's the hydrostatic pressure in the capillary. That's the glomerular capillary blood pressure. That we said was at 55. The plasma colloid osmotic pressure is a pressure that opposes it, along with the Bowman's capsule hydrostatic pressure, so that opposes. We can add those two together. That's 45. But since those oppose the filtration, we are going to subtract that from the one that promotes filtration. So we get 55 minus 45, or just go 55 minus 30 minus 15, that equals 10. And it's positive. So 10 millimeters of mercury of pressure actually promotes filtration. And that's what you see here. So again, hydrostatic pressure at the capillary is 55. We're going to subtract the colloid osmotic pressure from the capillary of 30. Okay, and we're also going to subtract the hydrostatic pressure of the Bowman's capsule that also opposes filtration at 15. That leaves us with 10 of a pressure that promotes filtration. So it's a relatively low pressure, but it works. It causes filtration at the glomerulus. Very important. Okay. So again, so that's the glomerular filtration rate. Okay, the rate at which fluid gets filtered. depends on these pressures and produces a glomerular filtration rate. So again, it depends on the net filtration pressure. Once you subtract the forces that are causing pressure to oppose filtration, that gives you your net filtration rate. Okay. It also depends on how much glomerular surface area is available. That's why the glomerulus is this big knot of capillaries to fit as many capillaries inside of there. And it also depends on how permeable the glomerular membrane is. And so remember, we don't seem to change these two, right? The glomerular filtration rate doesn't really change in a healthy body, and neither does the permeability of the membrane if you're healthy. If you get a breakdown, things happening if you always have chronic high blood pressure, that could break your very delicate glomeruli, right? So then you might change these two, and then you'll see a change in the urine output, right? And that's what medical personnel will look at. But mostly it's this change in filtration pressure that we can change the filtration rate, right? And so we can do that. We might have controlled adjustments in the glomerular filtration rate, because if you had a change in blood pressure overall, and we know we do on a daily basis, what we also know though is that the glomerular filtration rate, the GFR, doesn't really change too much in our kidneys. So the kidney must be doing something to adjust that, okay? And we have two major control mechanisms. We have a myogenic mechanism. Remember, myogenic refers to the changes in the smooth muscle, whether it constricts or dilates, of the arteriole. Okay, we're also going to look at something called tubuloglomerular feedback. I'll get to that in a minute. Let's look at the myogenic mechanism. And so, of course, I have this is autoregulation. The kidney, it's an intrinsic control of the kidney. This is going to happen automatically. Okay. Let's see. So here is a stylized view of the glomerulus. Here's the afferent arteriole coming in, and here's the efferent arteriole. All right. So what happens is if all of a sudden there is so before this happens, if there is an increase in blood pressure, right? Remember, an increase in blood pressure in the body is going to increase the rate of flow into all of our capillaries. Okay, so that will cause stretch on the walls of the afferent arteriole and each of these nephrons. And when it does, remember, what is the response to stretch? Is constriction. So this picture that you're seeing here is the response. So what that does is it limits blood's flow to the glomerular capillary. So when at first it went up, the vasoconstriction causes the blood pressure to lower, which then will maintain the GFR. So we might have had a momentary increase in GFR. This action of vasoconstriction will lower it back to normal. Okay? Lower it back to normal. Now let's say we have a decrease in MAP. Right? Remember, when there's a decrease in pressure, then that stretch is taken off of the smooth muscle of the arterioles. And when the stretch is taken off, its response is to relax and vasodilate. So again, first step it through. If there's a drop in blood pressure, then that means the glomerular blood pressure is going to go down. So the automatic response is the drop in blood pressure, vasodilate. That then is going to increase the blood flow, which is going to increase the pressure, which will raise our GFR, where before it was declining, now it's raising. So again, this picture is showing you the response. Okay, so the first condition here is a lowering of blood pressure. So the response is vasodilate, so that increases the blood pressure in the glomerulus, which allows the GFR to increase back to normal. Okay? So again, that is the myogenic mechanism. So the volume of filtrate produced by both kidneys per minute, it averages about 115 milliliters per minute in women and 125 milliliters a minute in men. Remember, men tend to have a higher amount of blood because they have bulkier muscles, right? That totals to about 180 liters of filtrate a day. That's 45 gallons that you filter a day, right? This rate stays relatively stable through a range of arterial blood pressure changes. So again, if you're exercising or something, this rate is going to remain the same. And that's partly because of this autoregulation, the myogenic mechanism. Okay, so those of you who are in lab, you're doing this in that PhysioX. You're going to manipulate the afferent arterial and see how that changes the filtration rate. You could manipulate the efferent arterial. We seem to rely on the afferent arterial, but we can manipulate the efferent. But so step it through. If we vasoconstrict the efferent, that means less blood flow leaving the glomerulus. If there's less blood flow leaving the glomerulus, then we're going to get a higher pressure in here, and that's going to increase filtration rate. Okay, so we might do that if we have a really low drop in MAP and MAP, mean arterial pressure. We might then constrict the efferent arterial to help raise pressure in here. Okay, vice versa, if we have a really, also our blood pressure is really high, then we want the blood flow to go faster through the capillary. That's going to slow down filtration rate. So we might vasodilate the efferent arterial. That's going to decrease the blood pressure because it's moving through and getting out of there. Okay? All right. The next one is this tubuloglomerular feedback. So that seems scary, TGF. Right? But just break it down. It must be something going on in the tube. The filtrate is inside the tube. We must be getting some feedback toward the glomerulus to manipulate what's going on there. All right, so let's step through. And we do. So that happens in this area. This is called the juxtaglomerular apparatus. What that means is it's a place where you have your Bowman's capsule with the glomerulus. That's called the renal corpuscle. The afferent and efferent arterioles are branching out, and this, notice this is the, go through your structures, this is the proximal tubule, loop of Henle, so the distal tubule actually twists around and comes in between the efferent and the afferent arteriole. So if we look at that, we blow it up, that's what we see right here. Here's the distal tubule. Right, here's the efferent arterial, here's the afferent arterial. And look, we have some specialized cells, okay? Within the tube, within the distal tubule, we have the macula densa. So these are the epithelial cells that are specialized into the macula densa. And these cells can detect the changes in salt level of the fluid that's inside the tube. Okay, so filtrate is already flowing in here and it's going to detect the changes in salt level. Okay, here, these are the granular cells. These are specialized smooth muscle cells surrounding the afferent arteriole. Okay, so these are called the granular cells. These are going to come more into play a little bit later. Okay, so all together, this little kind of this triangle area is called the juxtaglomerular apparatus. So basically, if your GFR increases, that means more fluid is flowing through the tubule, right? If you have more fluid flowing through the tubule, you likely have more salt, okay? So you have more salt in there. So as it comes through the distal tubule and the filtrate runs by the macula densa, those cells sense that increase in salt level, and it releases ATP and adenosine. ATP and adenosine. These are paracrines, okay? Local paracrines that will then cause the afferent arteriole to constrict, and that reduces GFR back to normal. Okay, so we're getting the same result as the myogenic response, but the myogenic response just reacts to blood pressure. Okay, this, the tubuloglomerular feedback, is reacting to what's in the filtrate. So if you have more filtrate, you have more salt. The macula densa senses that, releases ATP and adenosine as paracrines. That causes the smooth muscle cells of the afferent arteriole to constrict. It's going to lower pressure and slow down filtration. So then the macula densa will start to get less fluid and less salt. And so the paracrines will stop to be released, and that will open this up again. All right, so all of this just to control that rate in GFR, right? To keep it at a normal rate. So we're constantly doing these little adjustments, okay? If it's really low, let's say that there's less fluid in here and less salt, that meant the GFR went down. So the macula densa senses that, okay? It can release nitric oxide, right? That's a paracrine, that's a vasodilator. So that stops the action of ATP and adenosine, and that allows the smooth muscle to relax, and we go back to normal. Okay, so rewind that if you need to hear it again. All right, so that's the tubuloglomerular feedback. Those are autoregulation. Okay, the autonomic nervous system, though, can kick in. We have extrinsic control, right? Through the nervous system, that's extrinsic control. And this usually happens through the sympathetic system. This is aimed at more of a long-term regulation of arterial blood pressure. So the autoregulation, myogenic and tubuloglomerular feedback, those are mainly for, again, these small little adjustments that are constantly happening within the kidney, because our blood pressure is going up and down. Right? But if you have chronic high blood pressure or low pressure, then your sympathetic control will kick in. Okay, so that's the nervous system input to the afferent arterioles, and this is a baroreceptor reflex. Baroreceptor, remember, is a type of mechanoreceptor that's embedded in our blood vessels that can sense blood pressure. Okay. So if our blood pressure goes down, the baroreceptors sense that, sends the information to our brain, right? We get a stimulation of sympathetic nerve activity. So a lot of things can happen. Let's first go down the middle, because this is about the kidney. Okay, so sympathetic nerves then can vasoconstrict the afferent arterioles. There's nerves connected to them. They vasoconstrict. Right? We said our blood pressure was going down. We're going to reduce GFR. Stick with me. You're like, that doesn't make sense. Yes, we're reducing GFR and we're decreasing urine production so that we hold on to the fluid. So we increase the volume of blood, which increases pressure. Right? Remember, this is the overall long-term control. Overall, you have your MAP is down. Your blood pressure all over is down. Well, if our blood pressure goes too far down, then our kidneys won't function. So first what we do is we do a sympathetic activity and we lower GFR so we can bring our blood volume back up. That should reverse the blood pressure problem. Okay. If this is chronic, then this exasperates the problem, right? And we're going to always have a low GFR, and that might hurt our kidneys over time, all right? Exercise can do the same thing. You usually have lower urine production when you're exercising, right? So just step this through. We can add to this then when we have low blood pressure. Remember, we have other routes of sympathetic activity, and we increase cardiac output. Right? We might vasoconstrict to the skin and the GI tract, and that increases total peripheral resistance, everything that helps to raise blood pressure. Okay, so that's how we control it. That's the control, right? So I started to talk a little bit about this, unregulated influences on the glomerular filtration rate. So if you pathologically, you have plasma colloid osmotic pressure and the Bowman's hydrostatic pressure can change. So, so somebody who's like a severely burned patient, when you suffer severe burns, you expose the skin, right? You third degree burn will get down into, second and third degree burn will get down into the capillaries, right? And so they can lose large quantities of the plasma through the surface of the skin that's been burned. And remember, plasma contains the proteins, right? So that would reduce our plasma colloid osmotic pressure, okay? So if that happens, right, let me go back to this picture. We reduce colloid osmotic pressure, right? You can see this number is going to go down. All righty, let me go back so you guys can see. There we go. That means our GFR goes up, right? Because we're reducing the pressure that opposes filtration. So that means the hydrostatic pressure from the capillary has a bigger influence. So we'll go from 10 to 15 to 20, something like that. So that increases GFR. But again, too much GFR, we're going to get too much filtration. You could lose a lot of water, get dehydrated. It could also harm the capillaries themselves. Okay, dehydrating diarrhea. In diarrhea, you have less water in the body, right? With diarrhea, you're losing water through the digestive system, right? Through your number 2, through the anus. Sorry, I'm trying to be polite. It's anatomy, through the anus, right? So the water is being released, but the plasma proteins are staying. So now you have more plasma proteins. You've increased the colloid osmotic pressure. That means you have a bigger opposing force to prevent filtration, so your GFR goes down. Okay. What about this one, the Bowman's capsule, hydrostatic pressure? What if you had a kidney stone further down in your urinary system? Right? Sometimes they're in the renal pelvis, they're in the ureter, right? So what that does is that all that fluid, that urine passing through the nephrons, they come out the collecting duct and they get stuck. The urine can't pass, so it builds up. So the pressure builds up within the kidney and it builds up a back pressure against the nephron. Okay, so let me go back to that slide again, so you can visualize it, right? So again, you have a kidney stone down here, right? And so that's building up this pressure, the Bowman's capsule, because everything is a back pressure. The fluid won't drain out. So you have this back pressure. So maybe this goes to 25. And that opposes filtration. So you're not going to get as much filtration occurring, right? Which is there. That will decrease GFR. All right. So again, I mentioned this before, kidneys receive about 20 to 25% of the cardiac output. And so we need to constantly monitor the filtration rate to make sure that we're getting the blood filtered at the proper rate and returning clean blood back into the system. All right, so that's filtration related to the GFR. So the next step is reabsorption. And I am going to end the video here. I can turn off my laser. I'm going to end the video here, and the next part 2 will cover reabsorption.